Welcome everyone. Today we are going to learn about flip-flop movements of plasma membrane. Now, in my previous video, I have mentioned that lipids perform 4 different movements. That is why it is called mobility of lipids. One is rotation, flexion, and lateral diffusion that I have already mentioned in my previous video. Now, in this video, we are going to learn about flip-flop. At first, we have to know what is flip-flop movement. We know that plasma membrane is lipid bilayer. So there is 2 layers of lipids present in the plasma membrane. The inner layer of the plasma membrane is called cytosolic leaflet because it faces the cytosol. And the outer layer is called exoplasmic. Exo means outer. When lipid molecules that are present in the exoplasmic leaflet migrate from exoplasmic to cytosolic or vice versa, like cytosolic to exoplasmic. When lipid molecules migrate from one layer to the other layer, this is called flip-flop. In these 3 different movements, the lipid molecules actually move from one layer. So this migration of one lipid molecules from one layer to the other is called flip-flop. Now in pure lipid bilayer, we have to understand pure lipid bilayer do not contain any protein. Like natural plasma membrane contain lipid bilayer and protein. In my basic concepts of plasma membrane, I have mentioned that plasma membrane are made up of 2 lipid bilayer and proteins. But in case of pure lipid bilayer, it is artificially created. They don't have any protein. So no protein present here. No protein. Why it is important, I will explain. So at first, in pure lipid bilayer, this is called liposome. I have already mentioned what is liposome in my previous videos. Liposome. So in pure lipid bilayer, scientists found that 3 different lipid molecules are present in the plasma membrane. One is called phosphoglycerides. I have already mentioned phosphoglycerides. Then sphingolipids. And another category is cholesterol. So in the plasma membrane, there are 3 major group of lipid molecules present: phosphoglycerides, sphingolipids, and cholesterols. In pure lipid bilayers, in the absence of proteins like liposomes, phosphoglycerides and sphingolipids, these 2 kind of lipid molecules can show rotation, flexion, lateral diffusion, but rarely perform flip-flop movement. But in case of pure lipid bilayer, cholesterol can perform flip-flop movement very rapidly. Okay, so these 2 lipid molecules do not spontaneously flip-flop, but they can show rotation, flexion, lateral diffusions. But in case of cholesterol, it can rapidly flip-flop from one layer to the other. Now, what is the reason behind this difference? You have to remember the reason is the polar head group. In case of phosphoglycerides and sphingolipids, the polar head group is large. And in case of cholesterol, the polar head group is a single hydroxyl group. Now, why polar head group is important for this flip-flop movement? Because we have to understand that the outer portion of the cell is actually aqueous solutions. And also the cytosol is actually aqueous in solutions. Okay. Now the polar molecules actually interact with these aqueous solutions, polar molecules, but the inside of this 2 layer is actually contain the fatty acids that is non-polar in nature. Okay, so this inner side is actually non-polar and the outer side is polar. So the polar head group can interact easily with aqueous solutions, because aqueous solution is polar in nature also. Now, for this flip-flop movement, the polar head group has to migrate from these non-polar fatty acids. So it has to migrate from let's suppose this head group has to migrate from this non-polar region to the aqueous region. So this head group must migrate from here to here. Okay, that's why it requires energy. As I have mentioned that polar and non-polar do not mix. So to move this polar head group from this hydrophobic core or non-polar portion, it requires energy. But for cholesterol, the head group is just a single hydroxyl group, so it requires less energy. That's why they perform in the pure lipid bilayer, rarely perform flip-flop, but it can rapidly perform flip-flop movement. Now on a time scale, it has been found that these phosphoglycerides and sphingolipids perform flip-flop on time scale of hours to days, but cholesterol can perform flip-flop movement on a time scale of seconds, just seconds. Okay, now why I have mentioned pure lipid bilayer? Because in the pure lipid bilayer there is no protein. Now in the plasma membrane, natural plasma membrane present in cells, there are some proteins that helps these molecules to perform flip-flop, because flip-flop movement is actually has some significance in the cell. Now in the plasma membrane, there are natural plasma membrane, there are some proteins present that helps in flip-flop. These proteins are called translocator. So I have to remove this writing here. So in the plasma membrane, there are some proteins present that helps in the flip-flop movement. These proteins are called translocator. Translocator. One example of this protein is called Flippase. Flippase. Now this Flippase protein can move the lipid molecules from exoplasmic to the cytosolic leaflet. Okay, remember this. Another translocator protein is present in the natural plasma membrane, that is called Floppase, that can move or migrate the lipid molecules from cytosolic to exoplasmic leaflet. Okay, and there is another type of protein molecules is present translocator called Scramblase. Scramblase that can move lipid molecules from exoplasmic to cytosolic and also from cytosolic to exoplasmic. So in both direction it can move the lipid molecules. So this is called translocator, Flippase, Floppase and Scramblase. Now I will show two different events in the cell that are actually performed by flip-flop movement of lipid molecules. So let me remove this. The first event I will explain, that is called programmed cell death or apoptosis. Apoptosis. What happens during apoptosis? Let's suppose this is the 2-lipid bilayer. During apoptosis, phosphatidylserine, one phosphoglyceride is called phosphatidylserine, in short we call it PS. So phosphatidylserine are these lipid molecules are preferentially, remember this term, preferentially are located in the cytosolic leaflet, not in the exoplasmic leaflet. In natural situation, these phosphatidylserine are located in the preferentially located in the cytosolic leaflet of the plasma membrane. Now, during programmed cell death or apoptosis, they migrate from cytosolic leaflet to the exoplasmic leaflet. Okay, it moved from cytosolic leaflet to exoplasmic leaflet. Now, in the immune system, there is a phagocytic cells called macrophage. Macrophage recognizes this phosphatidylserine that is present in the exoplasmic leaflet, because macrophage can differentiate that in healthy cells, in the exoplasmic leaflet there is very little amount of phosphatidylserine present, but now something has happened to the cell. That's why the phosphatidylserines actually come out from cytosolic to the exoplasmic leaflet. So phosphatidylserine sends signals to the macrophage. Macrophage are some phagocytic cells that recognizes these cells, and it actually phagocytoses the dead cells to clear from the body. So this is apoptosis. Now scientists have found one protein molecule called annexin 5. Annexin 5. This protein can bind with phosphatidylserine. So this protein can bind with the phosphatidylserine. Now if we tag this protein with a fluorescent dye actually emit lights. Let's suppose we have tagged this annexin 5 with a fluorescent dye called FITC. FITC can emit green light. So then we can detect the apoptotic cells. There is a technique called flow cytometry, by which we can detect these apoptotic cells. So this is the significance of flip-flop movements that actually occurs in cells. Now another physiologic functions of flip-flop is that during clotting, blood clotting in platelets, the same thing happens. Phosphatidylserine comes out from or migrates from cytosol to the exoplasmic leaflets to activate clotting enzyme that is present in the serum. So clotting enzyme. So this is another example of flip-flop movement. Hope this video will help you. Thank you for watching, and always keep learning.